The last topic of the chapter has to do with modulation of protein activity. So one way in which a cell can regulate the amount of activity by a protein is through upregulation. That's the production of new proteins. So for example, a cell might produce more receptors, enzymes, membrane transporters. Alternatively, a cell can downregulate a particular protein. So it could downregulate, say, a particular receptor type. So this regulating of protein concentrations is an important strategy that cells use to control their physiological processes. There are also other ways of regulating protein activity. If we go back to the model of our enzyme, we talked about induced fit earlier, where we have ligand 1 and ligand 2. They don't quite fit exactly into the active sites, but due to non-covalent interactions such as hydrogen bonding, van der Waals forces, and ionic bonding, they have an induced fit in which the active site changes a bit. The ligand itself might change a bit, so then you have a nice fit. So then ligand 1 and 2 would then bind to these two active sites, and then there might be some sort of interaction between the two ligands. So for example, these reactants might combine together and form a product. So again, when these ligand and protein come close to each other, they interact through hydrogen ionic bonds and van der Waals forces. The protein binding site changes shape to fit the ligand more closely. Now, we talked about that earlier, but we're going to go into why why that might be important. One thing is that in terms of protein binding, there are different isoforms of proteins that are closely related proteins that will have slightly different affinities for ligands. So for example, you have adult and fetal hemoglobin. So these are proteins. The fetal hemoglobin has a slightly higher affinity for oxygen than adult hemoglobin. And so that might be, that may be due to a slight difference in the configuration of the protein. Some hormones are actually inactive when first synthesized, so these would be called prohormones. Portions are often cleaved off, leading to an active form. Some proteins also require cofactors such as calcium, magnesium, and iron. So again, hemoglobin would be one that would require a cofactor of iron. So again, let's talk a little bit more. So here's a cofactor picture from your book showing a cofactor. Here's the inactive protein. You have the addition of a cofactor, and now suddenly the protein is now able to bind these two ligands, ligand 1 and 2. So there are certain proteins that do need these. They require the presence of a cofactor, such as an ion, a small organic molecule, to then be active. Finally, we also have protein activation and inactivation here. So we have, for example, often certain proteins are when they're first formed, they're in an inactive form. Then there are enzymes that come along and remove portions of the enzyme, and then you have an active enzyme, or protein or enzyme. So the inactive forms are often identified with the prefix pro, like it could be pro something or other, pro a certain hormone, like proinsulin, and the other possibility is to have the ogen at the end. So trypsin is a type of enzyme, and trypsinogen is the inactive precursor to that enzyme. Okay, so protein binding, there's modulation. There are chemical modulators that bind to proteins and alter their activity. So first of all, you have antagonists. These are inhibitors that decrease the activity of protein. They bind to the protein but do not cause a response. So you could potentially have antagonists for example, a cell surface receptor, which is a protein for a particular neurotransmitter. If you have some of these antagonists in the extracellular fluid around the cell, then they might inhibit the action of the actual neurotransmitter. And there are different types of inhibitors. One type of antagonist is a competitive antagonist or competitive inhibitor. These are considered to be reversible antagonists. So, for example, you might have a neurotransmitter that would normally bind to a receptor on a cell, and then if you added a competitive inhibitor, this inhibitor would bind to that same receptor, but it wouldn't actually activate the receptor. It wouldn't activate it, and it would keep it inactive. But if it's a competitive inhibitor, this particular antagonist, it would bind, but then it could reversibly unbind. So it's binding and unbinding. So it's a little hard to imagine, but at this molecular level, things will bind for a little while and then unbind. And when it unbinds, of course, that allows the possibility for the natural endogenous neurotransmitter to then, say, bind with the receptor and then activate the receptor. Now, there are also irreversible antagonists. So these are considered to be non-competitive. And what they do is essentially bind to the protein so tightly that they effectively don't ever unbind. And so eventually the protein does get recycled and so forth. But for the life of that protein, it's pretty much bound. And so there are certain toxins, like certain extremely, snake toxins, certain certain types of mollusks in the ocean have certain toxins that are so incredibly strong at binding to certain proteins in your body that they just simply don't ever unbind, or at least they don't unbind in a fashion that reasonable amount of time. So they're considered to be effectively irreversible. We'll talk about these more in a second. There's also allosteric modulators. These bind reversibly to a protein at a regulatory site that's separate. It's at a different physical location than the ligand binding site, the active site. And these allosteric modulators can either be antagonists, meaning they would inhibit the activity of the protein, or they can be activators. They can increase the activity of the protein. And again, we'll have a picture of that in a second. And there are also covalent modulators. For example, many proteins are activated or inhibited through the addition of a phosphate group. So here's some pictures. A competitive inhibitor blocks the ligand binding at the binding site. So we have these active sites. Normally ligand 1 and 2 would bind here, but if we have this competitive inhibitor, it blocks ligand 1 from, say, binding. And that prevents this catalytic reaction from going forward if this is in fact an enzyme. Here's a picture of an allosteric inhibitor. It's a modulator that binds to a protein at a site that's distant from the binding site. So here the ligand would normally bind here, but here we have an allosteric inhibitor. So it binds to the protein. This then changes the conformation or shape of the active site, and now the ligand can no longer bind to the protein, so the protein is now inactive. And of course, the other possibility is that you would have an allosteric activator. So normally maybe perhaps the ligand would bind with a certain affinity, but once you have the allosteric activator binds, it actually increases the affinity of the protein for the ligand, and this will make the reaction go much faster. Yeah, here's the picture of the allosteric activator, and now here it's inactive, and now they're saying once the allosteric activator binds with the protein, this active site becomes much more has a much higher affinity for the ligand. Okay, so these allosteric modulators bind reversibly to the protein at a regulatory site away from the binding site. They change the shape of the binding site. They can decrease the affinity of the binding site for the ligand, or they can increase it. Okay, let's do a few questions. Downregulation refers to increased what? Okay. Protein binding to only one kind of molecule or group protein binding to only one kind of molecule or group of closely related molecules is called what? What's that? What's the word that refers to that? Here we have a chloride ion in an aqueous medium. These little Mickey Mouse structures are supposed to represent water. This large thing is oxygen. The two smaller ones are hydrogen. So which is the correct orientation of these water molecules around the chloride ion in solution? Here's a well-known example of allosteric modulation. This is the effect of pH, or hydrogen ions, on oxygen binding, the oxygen binding ability of hemoglobin. You could look ahead to chapter 18 if you want to know more about that. A decrease in pH causes blank and the affinity of hemoglobin for oxygen. Okay, that's it for the questions.